So today we're going to focus on nitrogen metabolism in a sense where we're going to talk about urea cycle. That's our pathway or animal's pathway that aims to detoxify ammonia, okay? And we're going to start, actually we're going to try to finish amino acid degradation, right? The reason why we're talking about nitrogen metabolism is, of course, in your diet, especially in modern day, you really like to eat meat. Maybe some of you are vegetarian, but most of the population will have meat regardless of it's beef, pork, or chicken, some kind of meat in your diet. And meat has a lot of proteins. So let's say this guy is protein, okay? Protein, how do we digest proteins, okay? So first thing first, we're going to use a protease. Protease essentially cuts peptide bonds, okay? So we're gonna have proteins, we denature them, we go and use proteases, degrade them into individually, these are the amino acids, okay? And of course, if your diet is very heavily dependent on amino acids or proteins, you're gonna be wondering how do you get your energy, right? So amino acids, usually we'll just take the amino acids and make your own protein. But let's say you don't have a source of carbohydrates, okay? So where do you get your central metabolites from, okay? You're going to get them from amino acids, okay? And how, right? That's what we're going to talk about today. So if you look, this is the TCA cycle, and there are some key intermediates, for example, oxaloacetate, fumarate, succinate, alpha-KG, and up on glycolysis, you have pyruvate. In between glycolysis and TCA cycle, you have acetyl-CoA. Okay, so these are the central metabolites. Okay, so those are the metabolites that occur in all organisms. Okay, and then you look. Okay, so it turns out almost all of these amino acids, they are going to degrade finally into some of the central metabolites. For example, you got alanine, glycine, cysteine, serine, threonine, tryptophan that can go to pyruvate. Okay. And you got other things that can go to oxaloacetate, other things that can go to fumarate, succinate, and a bunch of these that can go to alpha-ketoglutarate. Okay, so different amino acids, when you degrade them, you're going to get different things. Okay, right? And some goes to acetyl-CoA. Okay? But the key message here is that all amino acids, they are going to get degraded into central metabolites. Okay? So we're going to have pathways that convert all of these amino acids into central metabolites. Okay? Key message number one. If you take a look at proteases, proteases hydrolyze peptide bonds. Do you get any energy out of peptide bonds? No. Okay, so here you do not liberate ATP. Okay, so here is a simple hydrolysis. Okay, so no ATP produced. When you make proteins, when you make peptides, you will use ATP, okay? So that tells you that when you build molecules, a lot of times you use energy, but when you get rid of the large molecules into individual small molecules, usually a lot of times you don't get energy, okay? So in this case, proteases do not give you ATP, okay? So number one, here you don't get any ATP. So we know amino acids have what? Amino group, okay? They got amino groups. Okay? Now, how about central metabolites? Does pyruvate have amino group? No, okay? Pyruvate is an alpha keto acid that looks like this, right? So there's no amino group on here. That means that somewhere along the pathway you lost the ammonia, okay? You lost the amino group somehow, okay? So that's the first thing we're going to talk about, right? So the very first step for amino acid degradation that occurs to many amino acids is going to be the removal of the amino group, okay? So you're trying to remove the ammonia, right? So if you get rid of the amino group, then your amino acid becomes organic acid, okay? It just becomes a carbon-based molecule, okay? So then we can very easily to degrade. Now, so amino acid degradation usually begins with the conversion to the corresponding alpha keto acid, okay, by either transamination or the oxidative deamination, okay? So let's take a look. This guy is amino acid, okay? And this guy is going to be an alpha keto acid, okay? So the easiest way to go from amino acid to keto acid will be using this enzyme called the transaminase, okay? And essentially what it's going to do is it's going to use alpha Kg, okay? By the way, alpha Kg looks like this. So for those of you that don't remember, 1, 2, 3, 4, 5. Okay, that's alpha Kg, right? Now, the transamination, what it's going to do is it's going to swap the location of this amino group with this keto group such that you get glutamate this guy is glutamate and then the alpha keto acid. Okay, so essentially all you have to do is swapping where the amino group is with the keto group. Okay, so you exchange them. Okay, and this is transamination. Right, it's a transaminase. So a lot of amino acid degradation begins with this step. Okay, so you take whatever amino acid you got, you take alpha Kg and react it with the amino acid. You produce glutamate and you produce the corresponding amino acid. Okay, now remember we talked about glutamate dehydrogenase on Tuesday, and we said that in animals, the glutamate dehydrogenase primarily function in the reverse direction. Okay, it functions to the alpha Kg direction. Right, so this enzyme right here is the glutamate dehydrogenase. Right, and it's going to produce ammonia plus NADPH. So that's the transamination. There's another type of enzyme that essentially will do the oxidative deamination. In this case, you're not doing a transamination, right? So you don't have a amino receiver. Okay, so here I would call it, this guy is amino donor, right? It donates the amino group. Here's amino acceptor, so it accepts the amino group, right? Here, you don't have an amino acceptor, right? Because all you're going to do is you're going to use water to replace ammonia. The resulting product that we want is still an alpha keto acid. Okay, we still want to have an alpha keto acid. Now, if you take a look at this carbon versus this carbon, okay, this carbon right here, it's got a hydrogen on it, whereas this carbon does not. That makes this guy more reduced. So you're going from a more reduced to a more oxidized. Okay, so you have to do an oxidation on this guy. Okay, turns out in order to do the oxidation, what you're going to do is you're going to react it with FAD and you produce FADH2. Okay? And then FADH2 can then react with oxygen to form hydrogen peroxide, and hydrogen peroxide goes through catalase to form water and oxygen again. Okay? But understanding that this is amino acid oxidase. Okay? So it's different from transamination. Okay? Transamination, if you don't look at glutamate dehydrogenase, there's no net redox. Okay? 如果我们不看那个 glutamate dehydrogenase 这一步的话，你如果只看 transamination， 它是没有 reduction oxidation change 的，因为它拿一个 amino acid 加上一个 keto acid 变成一个 keto acid 加上一个 amino acid， 因为它只是换 functional group， 但是这边的话会有一个 oxidation。Okay? Make sense? All right. So those are the two major enzymes to do deamination, right? Removing amino groups. Okay, so ammonia is produced as the product. we're going to spend a little bit of time talking about transamination. Okay, now transamination is the most flexible reactions for amino acid, both synthesis and degradation. Right? Now, transamination is the reversible transfer of an amino group from an amino acid to an alpha keto acid. Now, this guy is going to use this coenzyme called the paradoxal phosphate. Okay? In short, I think this guy is called PLP. Okay? Paradoxal phosphate. Now, the equilibrium constant of transamination reaction is close to 1. Okay? Therefore, the direction of the reaction is dependent on the intracellular concentration of the product and the substrates. 合理吧？ OK， 因为 transamination 嘛，你看哦，这个东西它是 amino acid， 然后这是 keto acid， 它的 reactant 这边是 keto acid， 这边是 amino acid， 所以站在能量的 standpoint， 它们其实是差不多能量啊，因为你的 starting substrate， OK， 你的起始物是 keto acid 加 amino acid， 然后你的 product 又是 keto acid 加 amino acid，so 它们的能量没有差太多。Okay, so 它们的 KEQ 呢是 approximately equals to 1. Right? And because of that, the direction of the transamination is going to be dependent on the concentration of the substrate and the product. Very important, transamination is involved in both degradation and biosynthesis of amino acids. Okay, because all you got to do is do a transamination and you can go from keto acid to amino acid. Right? So in degradation, you can use this. In synthesis, you can also use this. Glutamate is the most typical or common amino donor, as glutamate dehydrogenase is the most important NH3 assimilation enzyme. Okay, 所以站在 nitrogen, okay, nitrogen 的 metabolism 上 glutamine 这两个会是最重要的东西。Okay, so a ratio between alpha Kg and glutamate essentially defines the nitrogen availability in this organism. 懂我意思吗 ？OK， 因为 glutamate 越多，就代表你这个 cell 里面的 nitrogen 越多。哎，那如果是 alpha Kg 越多 ，glutamate 很少，那就代表说你的这个 cell 里面的 nitrogen 不多，合理吧？可以哈。哎，所以最重要的 amino acid 呢，其实是 glutamate，OK，glutamic acid， 哎，因为它是number one nitrogen assimilation 的 enzyme，OK，glutamate dehydrogenase. Transamination facilitated by paradoxal phosphate, okay, and essentially the reaction looks like that, okay, so it's a amino acid plus a keto acid gives you an keto acid and amino acid. Now, paradoxal phosphate, believe it or not, it's another one of your vitamin B, okay. Now this guy is vitamin B6. Okay, so throughout this entire class, okay, 你可以回家跟你妈说，你知道 B1 是干嘛的，然后 like B2 可能你也知道，然后 B5、B6、B7 好像是 biotin， 对不对？然后 B12 是那个做 radical reaction 的 cobalamin, right? At the end of this class, you probably know all your B vitamins, right, what they're used for. Vitamin B6 is called the pyridoxine, right? So this structure right here. So if an organism takes in pyridoxine, and what you're going to do is you're going to make it into paradoxal by oxidizing alcohol into aldehyde. This guy's an aldehyde. Okay? And then you're going to activate it, okay, using a kinase, activate it into phosphate. Okay? So then this guy then is paradoxal phosphate. Right? Minor, okay? Now, as a coenzyme, primarily we use paradoxal phosphate. We don't use pyridoxine. Some other form of paradoxal phosphate, you might have pyridoxamine phosphate, where you take the aldehyde, okay, and do a transamination. So if I expanded this out, it's going to, this is aldehyde, right? So it's going to R, that's C, double bond, O, H, right? So this is an aldehyde. And what about this guy? If you expand it out, it's going to be R, C, and H2. So going from here to here. What is that? Transamination. Okay, it's kind of like a transamination reaction. So your pyridoxamine phosphate is the intermediate form in the transamination reaction. This tells you that your transamination reaction is facilitated using this cofactor, okay, or coenzyme. Right, we're good? Okay, PLP down. Now, usually your PLP or your paradoxal phosphate is attached to a lysine residue in the active site of the enzyme via a shift base. Okay, now, shift base, okay, what do you do? Right? 我两个不同图啦，就这两个图其实一模一样，这两个 molecule 一模一样 ，OK， 一模一样的 molecule， 我只是稍微 rotate 它一下，因为那个在 textbook 里面是长这样，但是我平常看的时候是长这样，从就是这个 phosphate 在这边嘛 ，OK， 然后这个是 aldehyde 在这边。So if you react this guy with a lysine, and you remember lysine has a terminal amino group, this is your protein. And this residue is lysine, okay? And lysine is going to be attached to your PLP through a shift base， 就是一个 aldehyde 加一个 amino group， 然后你会形成这样子的 structure， 这个东西叫一个 shift base。Transamination mechanism. You got this active site now where you have the enzyme-bound paradoxal phosphate. Okay, so it's going to look like this, okay? So this is the lysine, and, you know, this is essentially enzyme. Okay? And here, this guy is the PLP. So far, so good? All right, we're good? All right, now, this enzyme is going to take in your first amino acid. Okay, it's going to take in the amino acid, and you're going to have a nucleophilic attack or bind the amino group onto the shift base carbon. And essentially what happens is you're going to replace this nitrogen with this nitrogen, okay? Shift base formation 那个 detail 的 mechanism 你可以去看那个我们在做 glycolysis 的时候那个叫做 aldolase 对 aldolase 的 mechanism 会长跟那个很像，就是你会不断的 pronate depronate， 然后 eventually 你的那个进来的这个 atom 会完全 replace 掉这个 ，OK， 反正回去看一下。But regardless, essentially what you're going to do is you're going to replace the amino group from the lysine with the amino group from the amino acid. Lysine 嘛 ，lysine 原本是 blah， 然后这边 NH2 对不对？然后你的 amino acid 这边也是一个 NH2 对不对 ？OK， 所以你的 lysine 跟你的这个 amino acid， 反正它都有类似的这个 NH2 的 group，OK，and then 我们现在这边呢，这边已经说了，我们的 lysine 是接在 PLP 身上，OK， 那 lysine 因为它是 NH2 啊，所以 all I have to do then is 我用另外一个 molecule， 它有 NH2， 然后去 replace 掉 lysine，OK，so this is what you get. The lysine, okay, the lysine guy, lysine now is here. What the lysine can help is it's going to be able to help you do a deprotonation of your alpha carbon on the amino acid. Okay, so the lysine group will deprotonate the amino acid alpha carbon, and then the electron go onto the shift base, and the rest of the pi system here, that's what I mean, pi system, because you got resonance, so it's very stable. Okay, so the carbon ion is very well stabilized by the resonance into the pi system and the nitrogen atom. Okay, so the carbon ion is stabilized. Okay, so the reason why you can pull off this proton is because the resulting structure is very stable. Okay, 就是像我们之前讲，如果你今天要 deprotonate 一个这个 carbon 上面的 hydrogen， 你需要有 resonance structure， 对不对？就像我们说 alpha carbon 上面都可以被 deprotonate， 因为它有 resonance. Okay, now this guy's resonance is even better because you have a large pi system downstream to share 那个 electron. Maybe okay? All right. Hey, whatever. Okay. After you do that, then what you can do is then electron from the paradoxal, okay, the paradoxal ring right here, is going to use to pull the proton from the protonated lysine such that you generate this structure, okay? And then what you can then do is you're going to use water to replace the shift base, okay? such that you got the alpha keto acid as the product. What's the problem? Your paradoxal becomes paradoxamine, 对不对 ？Paradoxal 原本这边是 aldehyde， 但是现在这边是一个 amino group. Okay? So then we're going to do something to it. Alright? So the next thing is you're going to throw in another keto acid to form shift base with this guy. Okay? And then we follow step 1, okay, etc. And then essentially you'll kick out the amino acid at the end. Okay? 所以 PLP catalyzed mechanism basically 就是 shift base 这个 shift base 那个 ，OK， 不断的在换你 shift base 的 attached 的 atom. So this is the mechanism for transamination. Now, transaminase, okay, turns out is a very, very useful diagnostic tool for organ problems. 有没有去做过那个身体检查，然后测你的肝指数 ？OK， 有没有说什么爆肝？不，大学应该还好啦，但是，等你到那种30岁 ，like 你知道，你有时候去做个身体检查，就跟你讲说，哎呦，这个肝指数有点爆，就要小心一点哦。OK， 肝指数它在看的是什么东西 ？Turns out。OK， 你去医院在测肝指数的时候，它其实在测的就是你身体里面的 transaminase。OK， Why? Most amino transferase or transaminase, okay, they use glutamate alpha Kg as one of the two amino and alpha keto acid pairs involved. Okay, now two such enzymes are important in the clinical diagnosis of human disease. Okay, we're going to test the serum glutamate oxaloacetate transaminase and the glutamate pyruvate transaminase. Okay, so this is their reaction. Okay, so both of them take glutamate. Now the acceptor, okay, the amino acceptor could be oxaloacetate or pyruvate, okay, depending on whichever one. Now, these two enzymes are really abundant in heart and liver. Okay, all right, and turns out if you have liver damage, right, 所以你的 liver 如果被 damage 了，你的 liver cell 会爆开，然后你的那个 cell 爆开以后，这些 enzyme 就飘出来，大量的飘出来，飘到你的那个 bloodstream 里面。Okay, so then if you go to the hospital, take a little bit of blood, okay, and then you don't have to purify. All you have to do is assay, test the activity of this enzyme. Okay, if the activity is very high, very sad for you, you have liver problems. Okay, now you have the symptom of liver problems. I shouldn't say you have liver problems, but you have the symptom of liver problems. Right? So then the doctors will typically tell you maybe you should go check it out. Right? By like I don't know 超音波啊，或者是什么乱七八糟的检查 ，OK， 去看看你的肝是不是有发生什么事情。各位知道肝上面没有神经嘛，对不对？所以肝病你其实不太会有反应 until it's too late。OK， 所以肝指数是一个很 rough 的 estimate of 你的 liver function。OK。Turns out your PLP is not only used for transamination, it's also used for other reactions associated with amino acids, okay? So transamination mechanism we saw right here, okay? What you can also do is racemation, okay? So typically we have L amino acid, right? But what if you want to make D amino acid, okay? So then you can use this enzyme which uses PLP to do it, okay? We previously saw that you're going to have a deprotonation of the alpha carbon, okay? And once you do a deprotonation of the alpha carbon, that means that you can also do a reprotonation of the alpha carbon. Now, all you got to do is the reprotonation of the alpha carbon, you protonate it on the other side. 我们刚刚在看 PLP 的 mechanism 的时候啊，有没有？就是我们这边不是有一个，就是 lysine 它不是会过来做一个这个拔 proton 的动作吗？然后拔掉以后啊，你这边不是会形成一个 carbon ion 吗？对不对？你这个 carbon 它只有3个 group， 对不对？它有，这是1个 group， 2个 group， 3个 group，OK？、所以它是一个 anion。Now， 对于 racemase，OK，、就是说我今天要把这个 racemase 就是比如说 L amino acid 变成 D amino acid，OK？All I gotta do 就是在那个 carbon ion 的时候呢，我去把 proton 丢回去，只是我丢回去的位置不同 ，OK？ 我不要丢原本的位置，我丢另外那一面，它就会变成 D-amino acid. Okay, simple. Decarboxylation, you have these enzymes called the amino acid decarboxylase. Okay, and essentially what they do is when you form the shift base intermediate, okay, right here, shift base intermediate, instead of deprotonating, okay, 我不要做 depronation. What I can do is 我可以直接让它这边这个 carboxylate 掉下来，然后呢，这个 CO2 直接飞出去 ，right? So that's already the decarboxylation, right? Then what you can do then is once you form this intermediate, you can protonate. Okay, you can protonate this carbon using a proton from the solution. Okay, and then what you can do is use a shift base swap to swap it back. Right, 这是 lysine 嘛，原本这边会涂掉一个 lysine， right， 这边用的是 lysine， and then 你 lysine 再回来，然后把这个 shift base exchange 掉 ，OK， 你就会形成一个这个 decarboxylated amino acid。那 in general 它就是一个 amine。OK， decarboxylation for example。This is an amino acid. If you do a decarboxylation of this guy, you remove out this, and it gives you primary amine. Okay, primary amine. All right, so these are the mechanisms. So in detail, so I just went through all of those. Now, amino acid decarboxylase is actually everywhere, okay? Especially in microorganisms. Right now, turns out there are a lot of products, diamines, they are synthesized from bacteria. Okay, so 我们先看下面这边。Right. 你有没有听过一个东西叫施胺还是什么辅胺那种东西？听起来就很变态的东西。Right. All right. So these things are 4-carbon or 5-carbon diamine. Right. So 4-carbon or 5-carbon diamine. Well, first thing first, you should know that amines usually smell really bad. Right. Amines smell really bad. Now, 如果你今天在外面有那种动物的尸体，有没有？就是死掉的东西，它不是会有腐烂的味道，腐臭，就是烂掉的味道。那个烂掉的味道，很多时候是这个东西，就是这种4个碳、5个碳的 diamine. Okay, how do you make those? Right, you can use lysine. Okay, lysine, ornithine. Okay, these are natural amino acids. Okay, and you do decarboxylation, you generate this, and this smells really bad. Okay, 所以这个是 bacterial degradation of amino acid. And you generate these things called cadaverin and putrescine. Okay, now they form these really nasty products. However, cadaverin and putrescine diamines, they're very useful for industry, okay, as nylon. Okay, 所以你们这样很多用的一些 nylon 的 material, 他们都是拿 diacid 加上 diamine, 它可以去形成这样子类型的一个 polymer. Okay, 那这种 polymer 呢，这种聚合物就是 nylon. 那 nylon 就是 acid 加 amine 去做的 polymerization. So 这种东西虽然说它it smells really bad, but it's useful for biotechnology and chemical engineering applications. Right? So you can actually use diamine to make nylon. Right? So that's more of a application side. Turns out amino acid decarboxylases also participate in the production of many neurotransmitters. For example, GABA. GABA, if it looks like this, can you guys guess the amino acid that's used to make GABA? 这个东西是 GABA, 叫做 gamma aminobutyric acid. GABA 几个碳？4个碳，对不对？那它的 precursor, 如果我跟你讲这是 decarboxylation 出来的，它的 precursor 几个碳？5个碳嘛，对不对 ？OK, 5个碳 amino acid, 然后后面有一个 carboxylate 叫做什么？啊 ？glutamate 没错，它就是 glutamate。哎，所以 amino 这个GABA 呢，它就是用 glutamate 做出来的。OK, now glutamate you have, right? Glutamate. 那知道 glutamate 站在我们平常吃东西来说，它是什么东西？就是你常常会吃到， 尤其你去那种什么热炒店啊，一定会吃到。glutamate 是味精 ，OK，glutamate 就是味精，味精就是 glutamate。那有没有说过那个，或者是你们有没有 feel 过味精吃太多会头痛 ？OK，maybe you know it's related to your GABA. I don't know, maybe related 嘛，对不对 ？I don't know 啦，我不确定啊，but come on, it's very similar stuff, right? So glutamate is used as the precursor, one of your neurotransmitters. Dopamine, serotonin, okay, those are very prominent neurotransmitters as well. Okay, 那个我不知道他们中文叫什么啦，但是你可以去 Google 一下，你 Google， 反正那两个都是 neurotransmitter. OK， 那你仔细去看他们 structure， 那他们也会跟你常看到的 amino acid 有关系。比如说，当然你没有 amino acid 长这样啦 ，OK， 但是你有一个 amino acid 长这样。Your neurotransmitters are synthesized from amino acids. Okay, let's get back to the topic. We are trying to degrade amino acid. Okay, you're trying to get energy from amino acid. Right? 所以你的工作呢 ，as 你的 body 呢，现在要做一件事情是，我吃一堆蛋白进来，蛋白变成氨基酸 amino acid， 那 amino acid 需要被我代谢到 central metabolism 里面，然后代谢成能量。比如说进 pyruvate 或进 acetyl-CoA。那 ammonia 怎么搞 ？OK， 那我们不是说 ammonia 对人体来说是 toxic 吗 ？Which it is. OK， 你没事不要去搞什么 ammonia 来喝哦，真的会死掉。OK， 那 ammonia is toxic. OK, however, when you are degrading amino acid, it's inevitable that you will generate ammonia. Okay, so what do we do with them? Right? Turns out, animals have evolved pathways to detoxify ammonia. Okay? For example, birds and insects, okay, they evolve to convert ammonia into uric acid, okay, which is quite insoluble and can be excreted. Okay? So uric acid, apparently it's not very soluble, so you can condense them and check out how many nitrogens you got. 1, 2, 3, 4. Okay, ammonia is 1 nitrogen, so you can put 4 ammonia into 1 uric acid, and then you can poop it out. Okay, 所以那个就是鸟大便什么的，你也不要去吃它啦。OK， 因为就是你可能会中毒。对，为什么会去吃它Okay, now most mammals like humans, what we can do is we can convert ammonia to urea. Okay, urea. Now, urea, maybe you guys vaguely heard this term before. 它是一个 chirotrophic agent. Okay, 就是你今天如果有一堆 protein 出来是 insoluble， 我要让它变成，我要暴力的方法把它变成 soluble protein， 让它 denature， 然后变成一条 soluble protein， 加一堆 urea 下去就 OK。OK， 所以那个是我们第一次听到 urea。Okay, now urea turns out, okay, urea is a highly soluble molecule, but it does not contain ionizable group. Okay, why? Because these guys are not ionizable. Okay, so because it does not have any ionizable group, so it does not affect the pH of the media, which is a good thing. Now, urea, it contains 2 nitrogen per molecule, okay, and it is produced through the pathway called urea cycle. Okay, now, urea cycle turns out, urea cycle turns out to be the first biological cycle discovered. Okay, so even though we usually, when we talk about biochemistry, when we talk about metabolism, we usually talk about TCA cycle first as the biological cycle. But turns out the first biological cycle, okay, cyclic pathway discovered was the urea cycle. Okay, now it was discovered by the same guy that discovered the TCA cycle. Okay, Hans Krebs. Right, 所以同一个人啦，所以你可以想象他 maybe like 很厉害吧。Right? So urea cycle. Okay, so this is what we're going to talk about. Urea cycle was discovered in 1932, 5 years before the TCA cycle was discovered by the same person. Urea cycle uses energy, okay, that's a key. It uses energy to convert ammonia, okay, or ammonium, okay, to urea. The net reaction, I'm going to throw the net reaction in your face right now. The net reaction is you take a CO2 plus an ammonium plus 3 ATP plus 1 aspartate, okay, aspartic acid, okay, and 2 water. And you get urea, the ADPs, and 1 is AMP, and 1 is fumarate, okay? 这个是 urea cycle 的 net reaction, right? Now, fumarate can convert to aspartate with the following net reaction. You take fumarate plus ammonia directly becomes aspartate, okay? And then remember, AMP is equal to 2 ADP, 对不对? Okay? So the total net reaction, if I simplify it, you're going to get CO2 plus 2 ammonia plus 4 ATP gives you urea. 可以吗？可以哈。Urea cycle 本身啦，就是 Urea cycle 本身的 stoichiometry 是这个。But understanding that fumarate can convert to aspartic acid by taking in ammonia, and then ammonia can then be counted right here. Okay, so together total, you will consume 4 ATP, but then you can quench 2 NH4. Okay, huh? Maybe, okay, maybe, right? All right, so turns out this is the urea cycle. All right, this picture is in your book. Now, urea cycle is composed of mainly 5 enzymes. They are called the carbamonyl phosphate, ornithine transcarbamynolase, okay, argininosuccinase synthetase, argininosuccinase, arginase, okay, 这个 5 enzyme 记得一下, okay? Now, so let's start from the beginning, okay? Now, 来 amino acid 在什么地方 degrade? 在 mitochondria 里面 degrade. Why? I mean, come on, you're trying to get energy from amino acids, and the energy workhouse is mitochondria, okay? So, all right, so in mitochondria, you got amino acid converting to keto acid, blah, 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 and you excrete a bunch of ammonia, okay, or ammonium, whatever. So you got ammonia in your mitochondria, okay? So what you're going to do first is you're going to capture it into an organic compound. So you take ammonia plus bicarbonate plus 2 ATP, and you get this carbamonyl phosphate synthetase, so that's the enzyme, and you get this carbamonyl phosphate. 对不对？那这个也是我们之前讲到第5个 ammonia assimilation 的 enzyme。OK， 它直接拿 bicarbonate 加 NH4 加 ATP 就合出来了。OK， now this is done in the mitochondria. Okay. Now, what is also done in the mitochondria is you're going to take ornithine. Ornithine, it looks just like lysine, but 1 carbon short. Okay, and you're going to use ornithine transcarbamynolase. Okay, and you take the carbamonyl phosphate plus ornithine, and essentially you kick out the phosphate. You put these 2 onto here. Okay, so all you're doing is, this is a phosphate. So on ornithine, you have NH2, lone pair, attack carbon, this guy leaves. Okay, just like that, simple. Okay, and you get this thing called citrulline. Now, citrulline comes out of mitochondria, okay, and then citrulline gets activated by throwing AMP on it. So this guy here is AMP. Okay, you activate that first. What you're going to do is you're going to throw an aspartate to it. Okay, and in this case, aspartate, you're going to react this guy with this carbon. It looks crazy, I know, seriously. It looks like nothing that we've seen before. Okay, and you make this thing called the argininosuccinate. Okay, it's arginine plus succinate. Okay, how do I know it's arginine? This guy's arginine. And then this guy's succinate. This is arginine. Argininosuccinate. And then you got this argininosuccinase, okay? ACE, that means it degrades it. So you split it, okay? You split it, and you spit it out. You get fumarate plus arginine. Okay? So it turns out this is also the pathway to synthesize arginine in animals. 可以吗？可以哈。All right. Arginine, and as you can imagine, you are going to somehow get rid of this, right? This thing looks like your urea, right? So you got arginase, okay? So you hydrolyze this guy, and you spit out your urea, and you regenerate ornithine. Ornithine goes back to the mitochondria, and you start the process again. 可以吗？这个 pathway 记一下哦。Okay. You gotta remember the urea cycle. Okay. All right. So this is the urea cycle. Now, let's take a look where the ATP consumptions are. Okay. So you got 2 ATP consumed right here to capture the ammonia. Where's the other 2 ATP? It's actually right here, because you generate PPI, right? So you eliminate AMP. Right? So the other 2 ATP is in your argininosuccinase synthetase. Make sense? Right? So that's the urea cycle. Right? Now, the conversion of fumarate to aspartate, okay, so we know we generate fumarate. Now, however, fumarate can be used to capture another ammonia in the mitochondria through the TCA cycle to oxaloacetate, okay, which then can undergoes a transamination with glutamate. Okay? So we know fumarate can go to oxaloacetate, right? And oxaloacetate can do a transamination to generate aspartate. Okay, how do I note that? Because aspartate, right? 1, 2, 3, 4. 4 carbons. Oxaloacetate, 1, 2, 3, 4. 4 carbons. Okay, here's amino group. Here is keto group. Transamination. Okay, now you might be wondering, but whoa, wait a minute. Here, malate to oxaloacetate, you generate NADH. But then how come I don't account for NADH? And that is because NADH comes in here to use glutamate dehydrogenase to capture ammonia with alpha Kg to generate the glutamate. Okay, so NADH is balanced. 为什么我说 fumarate 跟 malate 是同一个 oxidation state？ 那为什么 fumarate 到 aspartate 会没有一个 net reduction 或者是 oxidation？ Okay, 不会有 oxidation 的原因是因为从 fumarate 到 malate 这边 malate 到 OAA 会产一个 NADH， 但是这个 NADH 呢会被 glutamate dehydrogenase 拿去利用再吃另外一个 ammonia 进来。Okay, 所以这个 ammonia fixation， okay, 这个 ammonia assimilation is done by glutamate dehydrogenase. 可以吗？ Okay, so that's how you go from fumarate to aspartate. Okay, now ornithine transcarbamylase, essentially just like I said, okay, what you're doing is you are going to transfer. You're going to first activate citrulline. You activate that by activating it, this here, into a AMP, okay, such that when you add aspartate onto this carbon, you can kick out AMP, right? So when you add this aspartate onto this carbon, you form this tetrahedral intermediate right here. And then what you get is when the electrons collapse back down, the AMP is eliminated, okay? And then you generate argininosuccinate. It's actually, I think, in a way, not too difficult, right? So this bond, you actually form a new bond between this oxygen with this phosphate, but minor detail. This is the regulation of urea cycle. Right? Now, animals have both long-term and short-term mechanism to regulate flux through the urea cycle. Okay, in the long-term regulation, the enzymes of urea cycle are produced in high levels in animals with high protein diets. Okay, so if today, for a long period of time, you keep on eating meat as your diet, then your body will have to transcribe a lot more of your urea cycle enzymes. So it's the long term. Now, short-term wise, you can have carbonyl phosphate synthetase is allosterically activated by N-acetylglutamate. Okay, this guy, if you have this, means you have urea cycle. Right? If you have that, you have urea cycle because it captures ammonia plus CH2, CO2. Right? Ammonia plus CO2 gives you carbonyl phosphate. Right? And this guy is allosterically activated by N-acetylglutamate, which is made by glutamate plus acetyl-CoA. Okay? What that tells you is if you have a lot of glutamate and you have a lot of acetyl-CoA, okay, then you will activate your urea cycle. Glutamate is a representative of cells' ammonia level, okay, and as one of the first steps of amino acid degradation is the transamination to glutamate. Okay, 所以我们说 amino acid degradation 第一步基本上都会是 transamination 去做 glutamate. Now, 如果你的 cell 有很多 nitrogen, 你的 glutamate level 一定会高。那你的 cell 如果有很多 nitrogen, 那你是不是会需要 activate urea cycle 来把你的 nitrogen 代谢掉？合理嘛，对不对？所以 glutamate 会有一个 positive 的 effect 在 urea cycle 身上。Okay? Now, this enzyme, okay, carbonyl phosphate synthetase, is also regulated by covalent modification, okay, the inactivation of the specific lysine residue. Right? Now, the detail of this mechanism is not completely understood yet, but just to tell you it exists, but the mechanism unknown. Okay? Very, very last thing for ammonia detoxification is ammonia transport. Right? Now, urea cycle occurs primarily in the liver. Okay, so in the liver, you got urea cycle. However, your other parts of your body will generate ammonia because let's say 你今天去 gym 去 work out 有没有？你去 work out， 那在你在做那个 intensive workout 的时候，你其实是你会拉扯到一些你的proteins， 你的肌肉 ，OK？ 那as you're degrading, you actually force degradation of your muscle cells, okay? And your muscle cells, some of it will get degraded. Of course, you will reassimilate the ammonia, but if you are going to degrade your ammonia in your muscle, you have to transfer your ammonia into your liver. Make sense? Right? Now, to do that, you cannot just transfer ammonia. Okay, ammonia 不可能在你血液里面在那边飘 ，right? 不 OK. So ammonia in your most tissues, okay, first get converted into glutamine. Glutamine is an amino acid, so it can fly normally in your bloodstream, and that's okay. In muscles, okay, you have your ammonia reacting with alpha Kg, making glutamate, and then glutamate can pass the amino group onto pyruvate to form alanine. And then alanine can be transported into the liver. Okay, so you have different carriers. In this case, carriers are either glutamine or alanine. 这是 ammonia transport. Okay? Now, 你可能会好奇说啊，为什么不直接 carry glutamate 就好？ So amino acid degradation. First of all, in your diet, you have essential and non-essential amino acids. Hopefully by now you guys know we have 10 essential and 10 non-essential. Actually, first of all, we can degrade all amino acid. Okay? 所以我们吃进来，我们 of course 可以 degrade all amino acid. Why? 如果不能 degrade 的话，你就 like 会 accumulate 或者是你会拉出来。Okay? 那I don't think you hear about like we poop out amino acids. Okay? So we don't. So we can degrade all amino acids. However, we cannot synthesize all amino acids. Right? So the ones that we cannot synthesize are called the essential amino acids. The ones that we can synthesize are the non-essential. Okay, so we'll talk about the synthesis of these later, but today we're going to be focused on degradation. Okay, now 这个 chart 啊，其实你们可以稍微留意一下跟你们讲一下 ，amino acid 其实价钱还蛮贵的。OK， 所以如果各位未来要去做 I don't know bio industry 或 whatever， 你其实也可以 potentially think about 一个 business in amino acid production。什么 amino acid 很贵 ？lysine 很贵 ，methionine 很贵 ，and tryptophan 很贵。OK， 所以这3个呢是价值蛮高的。For example， 我们之前博班的时候，我们实验室有一个 project， 就是在 aiming at 怎么样子想去 recycle 这些 amino acid。我们之前就有一个想法说 ，OK， 那我们可以去收集什么牛大便、猪大便这些乱七八糟的大便，然后把它的蛋白全部水解掉，那是不是就变成 amino acids？ 然后我再从里面催化，就是萃取出我要的那3个。那其实这就是一个很好的 recycle business。OK，recycle 就是把它萃取出来，对不对？那这些 amino acid 要拿来干嘛？动物饲料。OK， 养殖，你要养猪、养牛。OK， 那把大便纯化以后再给牛吃，然后就一直 recycle 嘛。OK，right？ 不然 recycle， 不然你弄出来一大堆 animal poop， 那你要怎么办？现代社会，就是 animals 太多，我们的 farming animals 实在太多，所以常常不是会有人讲一些，就是你有没有在 Facebook 上面看到说什么？如果今天全世界人都吃素的话，我们就没有 global warming 了。Right， 就是大家都会种树啊，然后不会有 animal， 就会不是那么多的 respiration， 那大概就不会有什么 global warming。Okay， 那 maybe，so turns out 就是现在一大堆 animal， 那一大堆 poop， 那你拿去做肥料，其实还会有很多剩下，那剩下的你就要可以去做一些 recycle 的 purpose， 那这就会是一个很好的 recycling project。Right， somehow recycle out the expensive amino acids, for example. Okay。Yeah, so, 再跟你讲另外一个 example, cysteine, okay? Cysteine, even though 这边是 non-essential, right? So we can actually make cysteine, right? But turns out cysteine 在全世界的 market 有很大的一个 portion， 在 40% 左右吧，是不是美国，日本一家公司叫做 Ajinomoto, okay? 就是味之素，可能有听过，做那个味精做很多的。Ajinomoto 呢，他们就是在做 cysteine 的 production， 那是用bacteria 去做。那他们的用意是在于说，很多早期的 cysteine 是从头发里面萃出来的，因为你头发里面有很多那个，我们记得上学期有讲一个特别的那个 polymer structural proteins， 它有很多 disulfide bonds，right？ 那里面就有很多 cysteine，right？ 那早期有很多 cysteine 的来源其实是Chinese people 的 hair， 就是那个大陆人他们有很多头发，然后去收集一堆头发，然后去用那个羟酸强碱去把它 hydrolyze 掉，然后把里面的 cysteine 萃出来 ，OK， 然后拿去 do stuff。So that's kind of nasty, but if it's pure, it's pure. Okay, 20 different amino acids means there are 20 different chemicals. 20 different chemicals, then you have at least 20 different pathways required to metabolize them all. 好了，那站在你们的立场就来，要记20个 pathway 哦，会不会太狠了一点 ？OK， 我们看一下 ，OK， 让我们继续看下去。20 amino acids， 更可怕的是 ，through evolution there may be more than one pathway that exists to degrade one particular amino acid. Okay, for example.Your pathway and E. coli's pathway for degrading one specific amino acid may be different. Okay, so turns out there are going to be a lot of different pathways that you can use to degrade amino acids. Now, these pathways primarily differ in the organism that they appear in, for example, humans versus E. coli, the conditions that they're most compatible with, for example, pH and temperature, that kind of stuff, or O2, okay, presence of oxygen.For example, we have oxygen, but there are a lot of bacteria that grow in the absence of oxygen. Also, the resulting central metabolites they produce, for example, some go to acetyl-CoA, some go to pyruvate, that kind of stuff, okay? So here we will only discuss some of the pathways for amino acid degradation with particular emphasis on how and where in central metabolism these amino acid is converted to.Particularly, we'll focus on the central metabolites, OAA, oxaloacetate, pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, okay, and acetyl-CoA, okay? Now, these guys, the ones in blue, we call glucogenic. What does that mean? That means that they can be used to make glucose. 为什么？因为它们可以做 gluconeogenesis, right? 这个 pyruvate 本来就在 glycolysis 里面 ，OAA 可以用 gluconeogenesis 的 enzyme 往回跑，因为它回得去 pyruvate。alpha Kg same thing， 跑一半跑到 OAA， 大家都可以跑到 oxaloacetate go back。Okay, so those are glucogenic. Acetyl-CoA, ketogenic, meaning it can only be used for making ketone bodies or fats. You cannot make sugars out of acetyl-CoA. Okay, that you should always remember. So let's start. All right, amino acids degrading to oxaloacetate. You only have two. You have aspartate and asparagine. Okay, 我们现在看一下哦 ，oxaloacetate structure 长这样，它是一个碳、两个碳、三个碳、四个碳 ，4 carbon molecule, and it's diacid. Here is one acid. Here is another acid, and it's alpha keto acid because this guy alpha, beta, gamma. Right, so it's alpha keto acid. Right. So if you go from aspartate, this is alpha amino. Go from amino to keto, transamination. Okay, 谢谢各位 transamination. Right? Transamination is going to use PLP, right, as a cofactor, and the amino acceptor is alpha Kg, and you generate glutamate. Okay? So this guy is aspartate. Right? Now, aspartate is related to asparagine, right? Because remember, asparagine, all it is instead of an acid, here is an amine, okay? So you can go from asparaginase, you hydrolyze this bond to liberate ammonia, you make aspartate, aspartate becomes oxaloacetate, okay? These two, very easy, all right? Pyruvate is going to be complicated. Pyruvate, the simplest one would be alanine. Okay, alanine looks just like pyruvate, except it's alpha amino instead of alpha keto. Okay, so pyruvate 1 carbon, 2 carbon, 3 carbon, alanine 1 carbon, 2 carbon, 3 carbon. Same thing, transamination, done. Okay, very easy. Now, turns out cysteine can also go to pyruvate, and in a way, you should think, it makes sense because 1 carbon, 2 carbon, 3 carbons. Okay, so it's 3 carbons. So all you got to do is get rid of the thiol, get rid of the amino group, you go to pyruvate. Okay, 所以 cysteine 也是去 pyruvate 的。好，来 ，without looking at 你的 slides， 还有什么 ？What else can you think of？ 跟 cysteine 长得很像的还有 serine。Okay, serine looks just like cysteine. Right? So of course, you can get serine, right? So serine, you do your dehydration, and you can switch up. You can cut out the ammonia. In this case, you're actually using an enzyme called serine dehydratase. Whatever. You just have to focus on serine becoming pyruvate. Okay. Turns out, glycine is absolutely related to serine. Okay, so we're going to talk about this in just a minute. Okay, but we'll skip that for now. Now, threonine can be used to produce acetyl-CoA and glycine. Okay, so threonine, technically speaking, can be both glucogenic and ketogenic, but of course it is primarily ketogenic because it produces acetyl-CoA. But it also produces glycine. And then glycine, 2 glycine gives you 1 serine, essentially. Also, you got tryptophan degrading to alanine. Alanine becomes pyruvate, focused on remembering where these amino acids end up. Okay, so those going to pyruvate, you got threonine, glycine, serine, cysteine, alanine, tryptophan. OK， 可以哈，这个应该还好，所以你要记得这6个会到 pyruvate。OK， 那你要是可以背起来的话，当然很 OK。那如果你背不起来的话，那你至少看一下 structure，based on the structure， 你猜一下它大概会去哪里。OK。Alright, so that was that. Now we're going to spend a little bit of time on glycine and serine, since there's an E here, serine metabolism. Now, serine and glycine metabolism involve this cofactor called the THF, tetrahydrofolate, okay? THF is an important one-carbon carrier in the cell. Previously, we talked about SAM as the one-carbon carrier. Turns out THF is used to make SAM. Okay, so one-carbon carrier, methyl carrier. A methyl carrier is SAM. THF is even broader. Okay, 所以我们之前讲 SAM 是 carry methyl carbon 一个碳的 methyl carbon。methyl carbon 是什么 CH3 嘛，叫 methyl。THF 呢 slightly different. THF 我们叫 one carbon carrier， 它其实可以 carry methyl、methylenol, what else? Formyl, I think you can do that too, right? So 它可以 carry 的是一个 carbon 不同的 oxidation state. Okay, 所以它会跟 SAM 有点不太一样。Alright, let's take a look. This is THF, okay? You can take serine, okay? And serine can essentially throw the CH2OH onto THF, and you form this thing called the methylene THF, okay? And then you can take methylene THF, you reduce it into methyl THF, and look carefully at what happened. Here, right here, this is CH2. This guy, CH3. So it's a reduction. Okay? All right? And you go from methyl THF to methionine. And methionine is used to make SAM. Okay, huh? Right? Now, in the degradation, what you can also do is glycine can do essentially what serine can do. Okay? 我们说 serine 可以把这个 CH2OH 丢到 THF 身上 right? Turns out glycine can do something very similar. You throw away the CO2, throw away the ammonia, the rest here is CH2. You throw the CH2 onto THF, and you also generate this guy. 所以它 THF 呢可以 carry one carbon, okay, one carbon, okay, so it's a one carbon carrier. So it turns out THF is used in a lot of different places. For example, you can use THF for the degradation of histidine, okay, methionine, right, and then we talked about serine and glycine. Looking from here, 这一步, what happened? Let's take a detailed look. You have one serine as the substrate going down. The other serine goes to methylene THF, okay, and you kick out a CO2. All right, so what happened is you have two glycine become one serine. 这个图看得懂吗？看得懂吧？因为其中一个 glycine 是直接当 substrate， 那你的口 substrate 是什么？口 substrate 是这个 methylene 的 THF 或者是 folate same thing， OK？ 那这个 methylene 的 THF 哪里来？它就是要靠 glycine 丢一个碳到 THF 身上，然后另外一个碳呢就变成 CO2， 就像我们刚就是下一个 slide 有讲到的 ，OK？ 所以 THF 呢可以 capture 这个 CH2，right？ 所以这个 CH2 在这边，然后你就把这个 CH2 再丢到 glycine 身上，就会变成 serine，OK？ 就等于多了这一颗东西，两个 glycine， 所以是两个 glycine， 一个 glycine 拿去直接做反应，第二个 glycine 把其中一个碳丢到 THF 身上，OK？ 所以你吃掉两个 glycine 做一个 serine，OK？ 两个 glycine total 是4个碳，一个 serine total 是3个碳，哎，所以你 kick out 了一个 CO2， 那就是在这边 ，OK？ THF, okay, tetrahydrofolate comes from, of course, folate, okay? So THF is tetrahydro, hydro standing for hydrogen, 1, 2, 3, 4. Tetrahydrofolate. Tetrahydrofolate comes from dihydrofolate, 1, 2. Dihydrofolate comes from folic acid, okay, folate. Now, folate, okay, if I expand folate out, Okay, this guy is folic acid. Folic acid is vitamin B9. 你又可以回家跟你妈说 vitamin B9 is used for making serine, okay, and one carbon carrier. Folic acid, it has 3 parts to it, and it has glutamate, para-aminobenzoic acid, and this other 2-amino-4-oxomethylpterin. 给你一个 example, right? Long time ago, before 我们有什么 penicillin 啊那种东西 ，OK， 我们早期啊，人类早期的 antibiotics 是一些 sulfur-based 的 compound，OK， 叫 sulfonamide，OK，sulfonamide。那 sulfonamide 为什么会 work？虽然它的效果不是很好，然后它的 toxicity 也比较高。那它会 work 的原因是因为它长得就跟你的 para-aminobenzoic acid 很像，有那么一点点了 ，OK， 还蛮像的。那这个 para-aminobenzoic acid 呢是 required for bacteria to grow，OK， 因为它们要拿去做 folate，OK，folate 很重要。那如果没有这个 para-aminobenzoic acid 呢 ，bacteria 就 cannot grow。Now，因为我们的 sulfonamide 这个东西呢，它长得跟这个把 para-aminobenzoic acid 放到 folate 上面的 enzyme 很像，所以这个会 serve as an inhibitor 去 block 这个 enzyme。那你的 bacteria 就不能做了，就不能做 para-aminobenzoic acid. As a result, your bacteria dies. Okay? Now, humans are not affected because para-aminobenzoic acid is not required for growth of animals. 可因为我们吃的 diet 我们是直接吃 folate， 植物里面就会有 folic acid， 那我们直接吃 folate， 所以对我们来说不是那么伤，但是对 bacteria 来说会伤，所以这是早期的 antibiotics. Okay? Next, we have amino acids degrading to alpha Kg, alpha ketoglutarate. Obviously, you have glutamate, glutamine. Okay? 所以 asparagine, aspartate 到 OAA, glutamine, glutamate 到 alpha Kg, 这个一定要知道。Okay? What you can also get is arginine. Okay? We saw arginine, you got this arginase, which is part of the urea cycle. And you produce this thing, ornithine. We saw in urea cycle. And ornithine, what you can do is, check this out, okay? What you can do, first, you do a transamination to get rid of the terminal amine, okay? Now, amine, 你做一个 transamination, what do you get? You get aldehyde. So, RNH2, transamination, R keto group, H. Okay, so here, 2 H's, okay? All right, now you get this glutamate gamma semialdehyde. What you can then do is, you don't like the aldehyde, so I convert the aldehyde into acid. It becomes glutamate. Glutamate becomes alpha Kg. What you also have is proline, okay? Proline, you always remember proline as that weird amino acid that's, you know, a circular structure. But turns out, proline has an uncatalyzed equilibrium between the oxidized and glutamate gamma semialdehyde. Okay, so go from proline, you first do an oxidation where? Here. Okay, it's an oxidation. Initially, it's a single bond. Here, it's a double bond. So it's an oxidation. And then, once you do an oxidation, guess what? This guy, shift base. So shift base, reversible. Okay, so it's formed between aldehyde and amine. Okay, so this guy split becomes this. Shift base 的 formation 是 between 一个 aldehyde 加一个 amino group. Okay, 那你的 amino group 在这边 ，aldehyde 在这边。Okay. 所以他们两个如果做 reaction, then this is what you get. And now this reaction is reversible, so you get rid of the shift base, then you form amino group and aldehyde group. Okay, maybe okay. All right. Histidine is complicated, so we're just going to jump over histidine. But essentially, histidine also becomes glutamate, okay, and glutamate becomes alpha Kg. Okay, so here. You got glutamate, glutamine that you should remember, okay? Histidine, you don't have to remember, but you should know histidine goes to alpha Kg. Proline, arginine. arginine you have to remember because it goes to ornithine. And then you should be able to convert ornithine to glutamate. Okay? Next, we have succinyl-CoA, right? Succinyl-CoA, turns out you got valine, isoleucine, threonine, and methionine. Okay? Now, primarily, these amino acids, they don't go to succinyl-CoA per se. They actually go to propionyl-CoA. Propionyl-CoA can go to succinyl-CoA through the same reactions that we talked about in alpha oxidation. You can go from propionyl-CoA to succinyl-CoA. Okay? Remember, you require vitamin B12 in the process. 记不记得? Okay? Right? So here you got methionine, threonine, valine, isoleucine. Let's look at each one of them individually. All right. You see this chart and you're just like, whoa, okay, what is this? All right, this is looking at branched chain amino acids. Okay? Now, you have to remember two things for me about branched chain amino acids. Okay? First, you go from amino acids to the corresponding keto acid. Okay? 所以 degradation 第一步 always amino acid to keto acid. 在这个 case. Okay? And then your branched chain keto acid is going to undergo a enzyme called the branched chain alpha keto acid dehydrogenase. Okay? 那它们是 alpha keto acid 就跟 pyruvate 是 alpha keto acid 一样，它们可以去做一个这个 CoA 的 acylating dehydrogenase reaction. What does that mean? Okay? So if you look on top, so transamination, and then you do a oxidative decarboxylation. So you kick out the CO2, you generate coenzyme A. Okay? You generate CoA. Now, after you generate CoA, then we're going to use beta oxidation. Okay? So, for example, if you look at this guy, okay, this is the alpha carbon, that's the beta carbon, right? So you're going to do oxidation, hydration, dehydrogenation, cleavage. Three features, okay? Three features for branched chain amino acid. Number one, transamination. Number two, alpha keto acid dehydrogenase. Number three, beta oxidation, okay? That's it. Three processes. Methionine degradation, okay? Methionine degradation. You go from methionine and you can go to homocysteine, okay? And this is actually quite simple because you just get rid of the methyl group. And how do you get rid of the methyl group? SAM, okay? You convert methionine into SAM, into SAM. SAM transfers the methyl group onto something, and then you get SAH. Remember that? SAH becomes homocysteine, and homocysteine becomes cysteine. Okay? 所以 methionine degrades to cysteine. 那其实 it should make sense because methionine is the only other amino acid that has a sulfur inside. 对不对？因为 cysteine 跟 methionine 是唯二有 sulfur 在里面的。Okay?